Now on to cell walls. Cell walls provide shape to the cell and protection from the environment. Bacterial cell walls are composed of peptidoglycan, which is modified glucose units, as you see here, which are attached to short peptides. Get it? Peptido for the peptides and glycan for the modified glucose units. So here's a diagram of glucose to give you some reference. The modified glucose units are N-acetylglucosamine, or NAG, and N-acetylmuramic acid, or NAM. See, on NAG, the hydroxyl group here on C2 is replaced with an amine group. On NAM, we have the amine group on C2, and we have a muramic acid on C3. So, NAG and NAM are strung together into long polypeptides. So these polypeptide chains are connected by tetrapeptides here and septa, I believe there's 7 peptides here. Together they make peptidoglycan, which, by the way, is only found in bacteria. Archaea have a wide range of molecules making up their cell walls, but they never have peptidoglycan. Now let's talk about the differences in structure of gram-positive and gram-negative cell walls. Gram-positive cell walls have a thick layer of peptidoglycan. All gram-positive peptidoglycan layers are studded with tachoric acid or lipotachoric acid, which is tachoric acid with a lipid group attached. Some gram-positive cells also have mycolic acid, a wax. Yeah, that's confusing. It's a wax with an acid group on the end. We call these cells acid-fast bacteria. Then we have some bacteria that have a thin layer of peptidoglycan sandwiched between the cytoplasmic membrane and another lipid bilayer called the outer membrane. The space between the two lipid layers is called the periplasmic space. Back in the old days, before electron microscopes, we couldn't see the difference between the cell structures, the cell wall structures. But we knew that there were two basic groups of bacteria, the gram-positives and the gram-negatives, based on how they reacted to a staining protocol developed by Hans Christian Gram. He would apply a purple primary or first stain, which we now know stains the peptidoglycan of gram-positive cells, so here, and the outer membrane of gram-negative cells. He applied iodine to make the crystals of the purple stain, crystal violet, bigger. Then he would use a decolorizer, an acid-alcohol mixture. Today we use 85% ethanol, which is less likely to eat a hole in your hand. Anyway, the alcohol would make the outer membrane more porous or soluble, allowing the crystal violet to wash away. The thick peptidoglycan of gram-positives resists this step, keeping the primary crystal violet stain. But as you will learn in lab, if you leave the decolorizer on too long, even gram-positives have to let go. The gram-negative cells are hard to see because they're clear. We add a secondary stain, a red one called safranin. So gram-positive cells hang on to the primary stain, are purple, and are positive for the gram stain. Gram-negative cells release the primary stain, are stained red with the secondary stain, and are negative for the gram stain. Nifty, huh? Now, I should mention that acid-fast bacteria, those with mycolic acid in their cell walls, stain weakly or not at all with the gram stain. We have to use a different stain, the acid-fast stain, to differentiate them. But we will talk more about that in the next chapter. Before we move on, Archaea come in gram-positive and gram-negative cells as well, but they have different cell wall structures than bacterial gram-positive and gram-negative cells. Still, the gram-positive Archaea retain the primary stain and are purple at the end of the staining process, and gram-negative Archaea lose the primary stain and are red at the end of the process. The last structure on the outside of a cell is the cytoplasmic membrane. Bacterial cytoplasmic membranes are mainly composed of a phospholipid bilayer. Phospholipids, as you may remember from Biology 201, have a hydrophilic or water-loving head, shown here as little round balls. They have two hydrophobic or water-hating tails, shown here. The heads interact with the water-based solutions on the inside and the outside of the cells. The hydrophobic tails are excluded by the water and turn toward each other. This arrangement is very good at keeping big, hydrophilic, and or charged molecules and ions out or in. It marks the boundary between the outside and the inside of the cell. There are proteins associated with this lipid bilayer. Integral proteins are embedded in the bilayer either partially or all the way through, like in this example. If they go all the way through, they are called transmembrane integral proteins. Other proteins are loosely attached on either one side or the other of the membrane. The fluid mosaic model describes our current understanding of the cytoplasmic membrane. Proteins can move laterally or this way, but not trans or flip-flop. Archaea have a different setup. They don't have lipid bilayers. Instead, they have one long lipid with an ether linkage in the middle and a hydrophilic group on either end of the lipid, a monolayer, if you will. It ends up performing the same function as a lipid bilayer, with hydrophilic ends interacting with the aqueous solutions on the outside and the inside of the cell, and a hydrophobic center to keep most things in or out. They have membrane proteins too. Before we discuss transport of molecules in and out of cells, let's review osmosis, or the movement of water past a semipermeable membrane. The cytoplasmic membrane is a semipermeable membrane. Water, being a polar molecule, doesn't move past the hydrophobic interior of the lipid bilayer very well. But the phospholipids are constantly in motion, and little holes, as represented in this diagram, are constantly forming and closing. Water can slip through. Ions cannot, since they hold hands with the phosphate heads, so to speak. Large molecules can't fit through these temporary openings. If there are more ions, think salt, or large hydrophilic molecules, think sugar, on one side than on the other, the water will tend to move toward the side with the higher salt or sugar concentration, like in the example in this figure. You can think of it as the water trying to dilute the solution until it's the same concentration on both sides. But we all know water doesn't have a brain, so it can't try anything. What really happens is water interacts with ions and sugars. The interactions, polar interactions with ions and hydrogen bonds with hydrophilic molecules, are kind of hard to break. So the water hangs out with the molecules that can't get past. The more molecules, the more the water hangs out. The more the water flows to the side with a greater concentration of dissolved molecules, or solute. That's osmosis. While we were at it, let's go over some definitions, shall we? The solution with the high concentration of solutes or dissolved molecules is hypertonic, like here in the diagram. The solution with the lower concentration of solutes is hypotonic. If they are the same, they are isotonic. So water will always flow toward the hypertonic solution. Now that we understand osmosis, let's talk about transport across cytoplasmic membranes. There are two kinds, passive and active. Passive transport may or may not use transmembrane proteins to transport the molecules in or out of the cell. It definitely doesn't use ATP or energy to transport the molecules. Active transport, on the other hand, always uses transport membrane proteins, and they use ATP to do it. The three main types of passive transport are diffusion, facilitated diffusion, and osmosis. Diffusion is the movement of molecules in a solution from an area of high concentration to an area of low concentration. For example, both CO2 and oxygen are nonpolar molecules that sort of dissolve in water. They can move past a cytoplasmic membrane like it wasn't even there. They don't interact with the charged phosphate heads, and the hydrophobic interior doesn't bother them. So if there is a higher concentration of CO2 being generated on the inside of the cell by metabolic processes, the CO2 will diffuse out without the cell needing to do anything. If the cells are using up O2, or oxygen, in metabolic processes, creating a lower concentration inside the cell, O2 will diffuse in. Now, sometimes diffusion doesn't happen fast enough, so the cell has transmembrane proteins that speed things up. For example, if water isn't diffusing in fast enough, bacteria can open up aquaporins to let more in, or out if that is what is needed. We've already talked about how water comes in and out through osmosis, which is a specialized type of diffusion. On to active transport. If a cell needs to bring molecules up their concentration gradient, or from low to high concentration, they will use active transport. For example, a cell will want to import sugar even when the concentration outside the cell is lower than the concentration inside the cell. That would be up the concentration gradient. This transport would be by uniport transmembrane proteins, which allow molecules to come in only one way, or out. Antiport is when a desired molecule is moved up its concentration gradient and an undesired molecule is moved out, down its concentration gradient. Think of it like as a prisoner of war exchange. You give us some of our soldiers and we'll give you some of yours, which are eating us out of house and home. Sometimes two desired molecules are moved up their concentration gradient, like in symport. And if two different transport proteins work together, it's called coupled transport. Lastly, there is group translocation, which only happens in bacteria. When a molecule, like glucose, is moved through the transport protein modifies it before spitting it out into the cell. In the case of glucose, the transport protein slaps a phosphate group onto it. Here are some diagrams of active transport. This would be uniport, this would be antiport, and this is coupled transport, with one transport protein moving molecules out and another one moving the ones that were just kicked out back in with another molecule, and this would be the symport part of the coupled transport, by the way. Whew, that was a lot. Well, the next topic won't be as long, since there aren't as many interesting, or I should say, easily visible things inside a prokaryotic cell as in a eukaryotic cell. Here is a summary of some of the different types of transport and some of the types of molecules transported thereby. This table includes eukaryotic cells, so this should be useful in topic 3.4. And here are the reminders of the general objectives as well, which I won't read again, but I will scroll through them so that you can take a look at them.